So thank you and welcome to everybody who is joining this webinar today. My name is Roger Tatoud. I am Deputy Director of the International AIDS Society, where I lead the Global HIV Vaccine Enterprise. Today this is the third panel discussion in our series of online events on design approaches for current and future HIV prevention efficacy trial.
Thanks, Roger, and a great pleasure to welcome you all. Those of you who've attended all of the sessions, you'll know that in many ways we're now coming to one of the most complex of the discussions, and that is really the design approaches for current and future HIV prevention efficacy trials. And today, to help me moderate this discussion, which we hope will really catalyze a lot of discussion in the field, because I think that's really what we need to be doing as we move into 2021. It's been an exciting year, 2020, for all kinds of reasons. But as we move into 2021, I think we have to be really applying our minds to how we will do this clinical trial business in the future. And to help me today, I have my good friend, Holly Janes, who will be helping to co-moderate and to manage questions and keep the discussion flowing. Holly, do you want to just say where you're from and what your line of expertise is? And then I'll work through each of the very exciting panelists to do the same.
Good morning, good evening. I'm Holly Janes. I'm at the Fred Hutch Cancer Research Center, and I'm a biostatistician by training and work with the HIV Vaccine Trials Network on vaccine trial design.
Good afternoon, good morning, good evening, and I'm sorry if my dogs are barking. I'm Glenda Gray. I am the co-PR of the HVTN, and I'm also protocol chair of 705, which is the Imbocoda, and also of HVTN 702 with Linda Gale Becker and Fatima and Moho, the vaccine trial that showed fertility at the beginning of the year. I'm glad to be here, and I'm also the president of the MRC. Thanks.
My name is Heisenmarker. I'm based in the Netherlands, working at the Janssen Vaccines and Prevention. I have spent 20 years in academia to work on HIV pathogenesis, and now with Janssen we have several vaccine programs, and amongst which a program on an HIV vaccine that we have 2 trials now for efficacy testing, the one that Glenda already mentioned in Bocodo, and we also have Mosaico, a study in MSM and transgender individuals in the Americas and Europe.
Hi, I'm Georgia Tamaris. I'm an immunologist at Duke University and part of the HIV Vaccine Trials Network. My laboratory is very interested in studying immune correlates of protection in regards to surrogates and understanding mechanistic correlates of protection.
Hello, everyone, and thank you for this very important session. I am a researcher and regulatory reviewer in the Office of Vaccine Research and Review in the Center for Biologics Evaluation and Research, or CBER, at the U.S. Food and Drug Administration. The majority of my regulatory and research portfolio has been on vaccines for HIV/AIDS, and I've been at the agency for more than 25 years. Thank you.
Hi there, Jeremy Sugarman, physician and bioethicist at Johns Hopkins. Been a longstanding chair of the ethics working group of the HPTN, the HIV Prevention Trials Network. I work on a variety of issues in ethics related to HIV, as well as a lot of other diseases and conditions. And it's nice to be a repeat offender in this series.
Yeah, thanks, Linda Gale. My name's Dean Fallman. I'm head of biostatistics at the National Institute of Allergy and Infectious Diseases. I've been there about 20 years, and I've been working on HIV vaccines on and off since I joined there. My interests lie in sort of statistics, clinical trial design, vaccinology, immune correlates. All stuff related to vaccine is what I'm happy with, basically.
Greetings, I'm Rob Newell, based in Atlanta, Georgia, where I serve as the Director of National Programs for the Black AIDS Institute, and I lead our Black Treatment Advocates Networks, our training capacity building, and all of our projects with the HVTN, HPTN, and the CoVPN.
Thanks, Rob. So I think you'll all agree a really diverse and exciting panel. There are others in the audience and around who also can make contributions, and we really hope you'll be sending your questions even as the discussion starts. We want to hear from you and interact with you as much as possible. So just quickly to remind, you've already heard that currently we have 3 efficacy vaccine trials in the field. On the one hand, we have Mosaico and Imbokodo, as you heard from Hanneke. This is the AD26 GP140 vaccine regimen, the one being conducted in men who have sex with men around the world, and the other amongst women who have sex with men here in Africa. You will remember, if you have participated, if you haven't, I hasten to encourage you to go and look at past versions of the design workshops. Susan Buchbinder presented a very nice description of Mosaico, where she described the layering approach to design. So this notion of combining PrEP as a standard of prevention, finding those individuals for whom PrEP doesn't work, and then enrolling them into a vaccine trial. I'm sure that will come up in the discussion as a possible and potential way of continuing. And I think I'm really looking forward to hearing from the panel its merits and demerits in 2020, 2021. There is, of course, also the PrEP-VAC trial. This is an interesting PrEP design, vaccine design, a combination of the two. So reminding you that that is looking at a mixture of DNA, MVA, vaccine candidates together with PrEP. And again, reminding you from an earlier talk given by Eugene in one of the early design workshops, that was very nicely described. So please go back there if needed. So the big question at the moment is just this ever changing standard of prevention. And, you know, if you've been at all watching the news, you'll know that it just got a little more complicated, with the good news that an injectable has been shown to be 90% effective, a 9-fold decrease in HIV acquisition among women in this part of the world, Africa, East and Southern Africa. And that was HPTN 084. Of course, cabotegravir long-acting was the agent. We've seen earlier this year that it also works highly effectively amongst men who have sex with men. We also had news this week that the phase 3 Empower 022 study will be coming to the field in Q1 of a monthly antiretroviral pill to prevent HIV. So that's using the islatravir molecule, only requiring a small pill once a month. And that study will be starting. In addition, lenacapavir, another long-acting injectable, also moving into phase 3 later, early next year. So there's no doubt that the field is becoming more complicated. And I will also add, of course, we do have AMP results coming to us before the end of the year. In thinking through how we could tackle this panel, we wanted to raise some potential scenarios. Because we think it is important to still look for that important preventive vaccine. And the pipeline is rich. There is much backing up that needs to come into phase 2s and potentially phase 3s. So let's take this scenario kind of approach to think through what might the agenda look like in the near future. And I want to thank Holly for giving some thought to this process. So scenario 1 that we want to present to the panelists and to yourselves is that this is the best case scenario. So we find vaccine regimens that are found effective enough, that are effective. And maybe that's a discussion in its own right. How effective does something need to be considered effective? And potentially a correlate is identified. So let's take 705 and 706. One of them shows really good enough efficacy, and we are able to identify a correlate in the other. That would be our best case scenario and one we want to discuss. The second scenario we want you to think about is the other end of the spectrum, the no efficacy scenario. So the vaccines are not found effective, those that are in the field now. There is no correlate, and we're kind of back to square one. We have a pipeline that's, you know, out there. We understand we need a vaccine. What do we do under those circumstances? And then sitting in the middle, somewhere in between, is scenario 3, where the vaccine has some modest efficacy, but we don't have a clear correlate. So we're sitting perhaps where we were sitting when the Thai results came out. The good news was with the Thai result, we thought we also had a correlate there, but let's take the scenario of modest efficacy and no correlate. So panelists, I'm going to take you back to the best case scenario. We want to be optimistic as we start. And that is where, you know, take, for example, 705, 706, you have two vaccine trials testing a vaccine regimen in 2 different populations. One is found to be efficacious, the other one confirms the correlate. How do we move forward? What is a future trial design that we might want to do in trying to either modify that regimen, improve on that regimen? What is, you know, or there's another non-neutralizing antibody vaccine that could possibly use that surrogate endpoint. What are some of the thoughts? And I'll ask any one of the panelists to volunteer what they think about in that scenario. Anybody want to leap into the deep end here?
Yeah, sure.
Great. Thanks, Hanneke.
I want to comment. So I think it's a scenario that we dream of, right? That we will have high efficacy and maybe also a correlate of protection. To further build on the regimen, so to further improve, I think it's very important to distinguish between a correlate and a mechanistic correlate, right? Because you see that with these different platforms that you can have different correlates and you do not identify exactly what is the true explanation for the protection. So that I think will need some more work. And indeed, that's what you already said. It would be really building on the mechanism of action that we get in these 2 studies. And I also see that there is a huge pipeline of potentially novel mechanism of actions, and that would not benefit from of course, from this approach. So I think it indeed opens up room for improvement of the regimen that would then already be efficacious, and also see whether it could work against even additional clades, additional risk populations, right? That's also very important work that would need to be accomplished. But there is then limited room for vaccine components or vaccines that would depend on a completely different mechanism of action.
Yeah, I guess I would just add, you know, this is like a great situation, obviously, and just to amplify on what Hanneke was saying, I think you could try and like improve on the vaccine just using the immune correlate as a signal, and if you improve that, then that might be enough to license you know, a vaccine in the same kind of class with the same kind of mechanism. And so I think you can move in that direction to some extent. But for vaccines with different mechanisms of action, it could be like this doesn't really quite apply to them. I don't know how you sort of bridge one mechanism of action to another. Just don't know how to do that. So I think it might be limited to a certain class of vaccines.
Yeah, I was going to say that as well. So, you know, with the same class of vaccines, what you'd want to do is the aim is to try and make it available. So we would want to see whether we require less doses, and try and think of ways of how to scale up. I think the biggest dream for us would be how do you scale up an intervention like this, but most importantly, how do we simplify it? A lot of these regimens are double primes, double boosts, they're 4 vaccines, and there are issues of durability as well. So it would be important to try and cut down on the number of doses, try and minimize the regimen so that we can optimally roll it out, and then obviously use that basis to bridge to other like vaccine regimens.
So maybe just, sorry, Jeremy, let me see you and then I'll pose another question. Jeremy?
Yeah, so it's a great discussion, and this is one of those nice problems to have in terms of thinking about why we're still at the vaccine business in the first place, right? So to say the heretical. We've had many years of looking at vaccines. We haven't been terribly great at finding something that works. We hope and continue to, and sort of back up and say, scientifically and ethically, what's the goal here? What's the game? And the game is really to provide a durable means of prevention of HIV that's going to be taken up, and to get at populations who may not have the luxury of being able to access services. And so with most viral diseases, the way we can get at this is through vaccines, but we need something that's not 55 doses or whatever the procedure is and how to do it. So I think deliberating before just launching would be say, what are we trying to achieve? We see efficacy in a study populations which are quite large, but it's still not effectiveness. So what are the effectiveness questions when we roll the vaccine out, and how can we tweak those vaccines to optimize them and ensure safety and effectiveness in a large population, as long as there's truly clinical equipoise about where we're headed with that next sort of candidate or dosing schedule. So I think even though we'd be jubilant, we'd have the big vaccine party. I think stopping at that step and saying, what's the end game here?
And also breaking down the correlates. So in this best case scenario with efficacy and with immune correlates, if it's one of the immune correlates that was identified in the preclinical model, that's actually an easier path, right? It somewhat validates that preclinical to human translation and suggests you can use that preclinical model to do those iterations and improve upon the vaccine. If it's not that, if it's something, a correlate that's new, something that we had not been thought of, or it's not in a preclinical model, that does make it more complicated, but it's also equally exciting because those things can then be verified in a variety of different models to down select those immune measurements and test them very specifically, mostly in a preclinical model, to understand if those were the particular immune responses that are responsible. And then you can build your strategies based off of those.
So I think this is really exciting. So opening the notion of going back to the preclinical. So I want to ask the panel, are there any populations where you couldn't do that extrapolation of the correlate? So, and I'm thinking here the special populations such as pregnant women, Glenda, children under the age of 5. Is there any place where a regulator would say, hold on, no, we're not going to pick up on that correlate as a way to bridge to these other populations?
Well, it would be nice to be able to bridge to kids who are exposed to breast milk transmission. And so we know that the best way to eliminate PMTCT is to eliminate breast milk transmission. And we know that a lot of women seroconvert while they're breastfeeding. And we also know that a lot of women are not adherent around 6 months, from 6 months of age of the baby. And so it would be a great mechanism it would be a great way to try and look at how to apply this in postnatal transmission, and to see whether you could do, if we had a good correlate, we could then do bridging studies into HIV exposed infants, and then do studies to evaluate whether we've been able to eliminate breast milk transmission in much larger community-based studies. Once we've established that there is immunobridging that correlates to the correlate that was found in adults.
Yes, correlates typically are used for bridging studies when you're looking at the same vaccine and wanting to go in different populations, if you have a good, true correlate of protection. But if the vaccine changes or there's modifications, then it is a lot more tricky, as has been brought up by Hanneke and others, that it's important to distinguish a correlate from a mechanistic surrogate. And so it's really mechanistic surrogates that can help you more if you're going to vary your vaccine platform in other ways. But if you're using the same vaccine and you do have an established correlate and you want to go to other populations, the elderly, the pregnant, whatever, typically the good correlate is the way to go.
Great, thanks, Carol. And I see a nice question from Peggy J. Always good to have Peggy at these design workshops. Hi to you. She asks, and I guess this is a burning question, I don't know if Georgia, having sort of gone through the Thai experience, how long would it take to identify a correlate of protection, mechanistic or not? And just to add to that, to talk through the steps for correlate validation that may be envisaged, so we can just think through timelines as we go. So I'm not sure who wants to pick that up.
I can at least start with the immunology piece, and then and then go to some others. At least for the current vaccine regimen, there was a lot of foresight by Janssen and the HVTN to go ahead and begin thinking about this ahead of time. And again, qualifying and validating the necessary immune assays to plan the correlates analysis, and now to begin preparing the reagents ahead of time, in a sense, planning for success, such that when the results are known, you have all of the details in place, ready to go to perform the analysis, which will make it much faster. There's, of course, the pilot phase, generating the data, making sure it's reproducible across the laboratories that are running the assays, working with the data management and statistics team to get all of that in place and implemented, such that you can quickly go ahead and do the analysis once you have all of the data sets. So I don't know, Holly and Dean and others should comment on timing of analysis, but it's, I think it's within months to a half a year from the time that you know the result, where given all the pre-planning.
Yeah, I think you're right, Georgia, that it can proceed quickly like that. And I would just like to amplify on what's been sort of talked about here with mechanism versus non-mechanism. And I think what's supportive of a true correlate or a mechanistic type correlate is if you can sort of see a similar signal in the non-human primate model as in the human primate, as in the human in the field studies. One advantage of the preclinical models is that you can vary dose and thus vary the intensity, the immune response. You can calibrate that. And with a successful vaccine, vaccines you always try and have like way more antibody or whatever immune response is necessary. So it can be difficult actually in field trials sometimes to get a correlate. You rely on breakthrough cases and you rely on sort of the immune response being less than you would hope for ideally. But I think the more different bits of evidence you can get is what's required really to support a strong correlate that allows bridging and transportability.
So to push you a little harder, Bill Snow asks, don't you need a putative surrogate to begin with? And do we have one, for the current AD26 program?
I think that makes everything an awful lot easier and makes it more transparently legitimate or endorsed in a way, because otherwise there's a danger, you're sort of looking for different things. There's multiplicity issues where if you look at a lot of different correlates, you don't know what you'll find. You have to do a statistical correction for that, so it's less powerful and so on. So by far the best direction is to have a good guess or a good idea based on preclinical data and understanding a mechanism, and focus your analyses on that, and also like at a particular time and a particular measurement.
So before we leave the different populations, Peggy asks, is it not possible to have the expansion to other populations based on preclinical, say, tox testing and or safety studies in that population? So, can we get by without the need for this sort of surrogate application, but actually just base it on safety studies?
Well, I mean, we've tried before, Linda Gale, with adolescents and young infants, and so I think that in terms, maybe Carol would also speak about the regulatory concerns. So I guess obviously safety is important, and the regulators have been very, very conservative post Pambili and STEP to allow us to go into more vulnerable populations without establishing efficacy. And possibly once we know the outcomes of Mosaico and Imbocoda, this will give the regulators a much more pause to accelerate work in much more vulnerable populations. But certainly post Pambili and STEP, there was a lot of hesitance to work in adolescents and younger children.
Carol, I don't know if you want to take up that challenge. You're muted.
Yeah, I think we would definitely want to see immune responses or, the potential correlate of surrogate if you're going to bridge to another population. I don't think you could do it on safety alone. And it's especially important to know whether those different populations will respond the same way. So it could be a correlate, even if it's not a mechanistic surrogate. Obviously, mechanistic surrogates are better, but we would need to see an immune response.
One thing that I would like to add is that for the bridging between groups that have different routes of HIV transmission, there you already need to bridge. And another bridging that we need to do is, of course, from going from clade C dominant areas to other clade dominant areas to see whether that same correlate holds up. So between Imbokodo and Mosaico, we are planning to do sort of cross-check, right, whether the one correlate is also coming up in the other one, but to go to drug users or even other clades, we really need to get additional hands on whether these protective mechanisms can be translated to these other variables. So yeah, I agree with everything that has been said, but there especially in HIV, there are of course a lot of additional complexities that we need to cover.
I'm going to ask Holly if she feels that the last comment from Hanneke sort of answered that question around what is needed to validate correlates. It may be more complex. Yeah, you happy? Okay. So perhaps then let's change gears for a moment and go to the unhappy scenario where the current efficacy trials don't turn up the goods. You know, we've seen a very happy scenario with COVID today, this last week, a very different, outcome. But what we could find, and we hope not, but it could be that we don't have the efficacy we were hoping for. How does that change what we do, how we go forward, given how our prevention landscape is looking at the moment? Anybody want to jump in there?
I mean, we'd have to go right back to the toolkit again and try and design a vaccine that had a different strategy. And so up until now, we've been looking at non-neutralizing mechanisms, and hopefully one could look at other novel vaccine designs that we're doing at the moment, looking at B-cell lineage and trying to induce neutralizing antibodies in different ways. But we would, given the landscape, the treatment landscape, and how highly efficacious it would be, we obviously would have to convince the funders that there is merit. And certainly there is merit because we do need variability. Some of these interventions may be very expensive. So even though we've heard about 1084 and all the other studies, we have no idea how much that's going to cost us in the developing world, and we don't know when that's going to be licensed with us. And like COVID vaccines, we might be the very countries that need the long-acting antiretrovirals, we may be the last people to get it.
Yeah, thanks, Glenn. I'm going to actually ask Rob to comment here. It's the fundamental question. If we find ourselves that, in the next few months that we don't have, or maybe it's even a year before Hanneke can talk to our timelines, but maybe some time, but we find we don't have an efficacious vaccine. Should we continue the quest?
I really think it goes back to a question you hinted at very early on, which is, what is an efficacious vaccine? And I'm becoming more concerned that what research might consider an efficacious vaccine is not going to be acceptable to the public, particularly when we get these off the wall results like we've seen with COVID, like we've seen with long-acting injectable PrEP. Folks are expecting 90-95% efficacy, and that's not, I think, what we've been working toward. We would accept something less. Like a year ago, we'd have accepted something less than that and been very, very happy in the HIV community. And so I'm concerned that what is efficacious is different.
Yeah, so maybe, thanks, Rob, to put that to the panel. I mean, where are we sitting at the moment? What would we consider, and what are the implications of moving forward, to try and improve on a vaccine? What is a reasonable level of efficaciousness?
If I can add to what was just said too, because we have the AMP trial results coming out, which will tell us about a broadly neutralizing antibody approach as a proof of concept for a vaccine, and gets at what Glenda was talking about with this very strong pipeline underneath these current vaccine regimens that are building strategies to induce broadly neutralizing antibodies and work across and have strong T cells that can also be involved in protection against acquisition. And so there is a robust pipeline of ideas and manufacturing that would lead to, if there's a negative result here in a non-neutralizing antibody and no correlate, people would be leaning more on this pipeline and the novel ideas. I think the results from the AMP trial will help tell us if that proof of concept is right for a neutralizing antibody. The COVID vaccines also give us that answer too. If you can elicit a neutralizing antibody response, you might see high levels of protection.
And again, just to remind the audience that, you know, the antivirals, whatever happens, you have to have substance on board for them to work. The difference being with a vaccine is obviously you get the immune system working once the substance, you know, it doesn't have the same principle of needing that ongoing adherence. And to remind, again, everyone, not everybody's going to want an antiretroviral as an option. And also we are still to figure out what long tails might mean for safety and, you know, and futures for those individuals. In addition to which, you know, the antiretroviral target also becomes a problem when we see antiviral resistance come about. Cabotegravir long-acting is it's a cousin of dolutegravir, which is currently being rolled out around the world. And we know that antimicrobials do have a limited lifespan due to resistance, no matter how high the bar for resistance is. Ultimately, unfortunately, we find a way to, you know, to result in resistant mutations. So these are all important considerations. So I couldn't agree more, even as somebody who cares a lot about antiretroviral-based prevention. The place for a vaccine is really important, but it raises the question, and maybe that's something to pose to all of you, is what is the design of the future? Do we think the mosaico layering kind of approach, just to remind again everybody of the picture Susan Buchbinder showed of the mosaic, and, you know, finding those individuals as we continue, as we add cabotegravir to the list, and potentially islatravir and lenacapavir down the line, how big is the piece of mosaic that's missing that we can insert vaccines into? So in other words, will there be those individuals where antiretroviral-based prevention is not going to be an option for them, and they will then be able to volunteer for a vaccine trial? I just want to hear, is that layering approach still an option for us in 2021? Jeremy, I see you burning to answer.
Actually, oh yeah, I got my seatbelt on just so I don't jump out of this. I think, would you just make a clarification for me and for everyone? So my understanding from Susan's really nice presentation about Mosaico was that the layering approach permitted participants to access PrEP that's available, whatever PrEP services, versus something like 083 or 084, which had a double dummy design. So we're slightly different things. So I want to just make sure everybody was on the same page before we went there. Because if we have an existing standard of prevention that people are accessing, it's not really the same case as people who don't want to access. I mean, some will, but it should wash out in whatever randomization is there, right? So the problem you're going to have there is the same problem of the size of the trial necessary to, speaking without a statistics license. So I'll let one of you guys say this. Say the same thing, was basically the trials will have to be extraordinarily large in that setting because of the background conditions and presumed uptake of PrEP and preventive measures, so you won't have enough outcome measures. So that's the worry, I think, with more and more rollout of effective measures. I'm not ethically troubled by most layering designs, assuming that people actually can have access and that the standards of prevention locally are not somehow compromised because of other factors, like as a setup for a trial. But I'd be curious to see what others think.
I mean, coming from South Africa with such high HIV incidences that we saw in 702, you know, I would want to make sure that people could access the best prevention. You know, we have an epidemic that's out of control, and you know, my desire would be to to close that transmission gap and to make sure that that people who are on our trial, so yes, it takes bigger studies, but it's hard to not consider the use of long-acting antiretrovirals given their extreme efficacy. So that would have to be part of the equation. We can't, particularly in South Africa, when we have incidences over 4%, it's hard for us to continue to have suboptimal prevention strategies in our studies.
Yeah, but I just want to remind people of the data we saw this last week that, in the 20 odd sites that participated in HPTN 084, the long-acting cabotegravir in women who have sex with men, incidents across the board, and remember that's a double-blind, double dummy, everybody got something, whether it was a daily oral pill or a long-acting cabotegravir, and it was a superiority design. Across the board, the incidence there was 1%. So definitely saw impact in both arms there. That was significant. So just to remember that. Rob, you were going to chime in there. I cut you off. Yeah, I'm sorry.
I was just, I don't think that a vaccine study can be the reason that communities get access to appropriate prevention, right? We need to work on access to prevention, absolutely. But when that vaccine study leaves, so does access. And so that can't be, that's like having your healthcare tied to your job. Like we don't want to do that. So I think standard of care in the community, whatever that is appropriate to study the vaccine in those settings.
And actually, so Rob, it's a great point. And actually, if you go to the HPTN ethics guidance document, we lay out criteria of standard of prevention to articulate the notion of having effective means, making sure they're practically achievable, and making sure that they're reasonably accessible. So all three legs of that stool are important to understanding what's really there. It's a totally different setting than one that's set up artificially in the context of a study to study the question, right? I think I'm on the protocol team for 083, and really we labored long and hard about the double dummy design, the idea that we had to do something in that setting. But I think things are going to change over time, and this Mosaico is a nice transition state, I think, to thinking through what our ethical obligations are.
So, I wanted to just comment on a point Rob made, which was, COVID-19 vaccines have 90% efficacy or better, and PrEP also has very high efficacy. And so I guess, there won't be a lot of appetite for a vaccine with an anticipated efficacy of 50% or something. And it might be hard even to, conduct a trial for that. A sort of upside in a way is if we're only going to proceed with anticipated very high efficacy vaccines, the numbers work in your favor. Basically, you can have a relatively smaller sample size or trial to try and identify a vaccine with 80% efficacy, say that compared to 40. It will still be very large and maybe at the edge of what's doable, but I don't know, those comments made me think differently. Basically, that it's not like we need a proof of concept or something that shows that ultimately we need something that's super efficacious, or it just won't be taken up.
I think it's a public health benefit and a personal benefit, and there certainly will be a public health benefit for a 50% or 40% efficacious vaccine, and one could argue about the personal benefits, and maybe that's where access to other interventions are needed. But certainly one can't argue against the value of a less efficacious vaccine at a public health as a public health benefit at a community level. So although so I think that there still is value of a partially efficacious vaccine, and we should understand that it will impact on population transmission.
Yeah, I guess that's true. If it's taken up, and they wouldn't take PrEP otherwise.
So it's a segue to this notion of population-based sort of approaches, and I'd love to hear from the panel, whether that is potentially another way of going. Does that give us a potential loophole here? And the notion being that you approach a whole group community and offer them one intervention, and the background prevention is as it is, versus another whole community who doesn't. So what are the thoughts about that sort of design? Jeremy, does that change the sort of ethics in any way?
So, if you're talking presumably about something like a cluster randomized trial, right? So, the cluster randomization doesn't take away all your ethical issues. It just changes them, just like the same, same thing statistically, right? It doesn't solve the problem. It actually makes them more complicated. So if you think you have trouble speaking statistics in a usual professional meeting, go to one where they're talking about a cluster randomized trial, and you'll be hearing about the interclass correlation coefficient and all of these wonderful things that statisticians talk about and say, I'm not sure if we really know what the answer was there. So I think we have some huge methodologic concerns to start with. So the first thing would be, for the scientific question being posed in that cluster randomized trial or CRT, are the methods suited to answer this question? Because if we don't have scientific integrity and value in what we're going to learn, that trial has got to be informative. I mean, if we've learned nothing else in the history of HIV-related research, is that we have to learn from everything we do with people, right? And with specimens. So if we're not going to learn something from that trial, it's not ethical to do it. But assuming that you've got it up and you really believe we're going to learn something in this trial, regardless of what it is, cluster randomization is going to raise questions are, can you get community buy-in for to being randomly assigned? Presumably what you're talking about, and correct me where I'm wrong, I'm sure I'm wrong, but it's just where I'm wrong, that you're going to give some communities the placebo vaccine and other communities the active vaccine, and they're going to be randomly assigned to one or the other. Presumably the standards of prevention that are there are going to wash with the randomization if you do your cluster randomization properly. So I'm not sure what you gain necessarily. In addition, what we've seen, at least with some other vaccines, I know there's been quite a little bit of kerfuffle over the malaria vaccine rollouts that have been tested, where WHO gave the equivalent of a conditional approval to a vaccine. We're worried about the malaria vaccine and how it would roll out, and then did a cluster randomization for implementation science. And that caused an enormous set of concerns about how do we think through this issue. So if anything, what I would say is later stages, that scenario one we were talking about, you want to tweak something, maybe you could do that. But early phase, before we have something that's known to be really good, or at least partially good, it's going to be hard. It's a hard sell. And the amount of effort that went into 071 pop art, huge amount of effort to do a community randomized trial, how to collect HIV data. This is not, HIV is still stigmatized. The collection of data, the effects are going to be really monumental. So figuring out how the quality of the data we're getting is going to be tough. So sorry, you just hit a little out. And there are also ethics standards in there that can be brought to bear that complicate things.
Yeah, I would say it's difficult too. Yeah, just that you need a lot of communities, maybe I would like 20 to 40 probably. And I think the big problem is you need pretty much everyone in the community get the vaccine or the placebo, and how is that going to happen? I mean, that seems difficult. And the communities have to have a sufficient attack rate. So I think maybe superficially it seems like an advantage compared to the individually randomized trial in that small space of people who don't want PrEP and think vaccines might work for them. But where are the communities that would allow it to actually be feasible?
So I see thanks, Dean. And I think, thanks, Jeremy. I think we needed to discuss that as a possibility, because I think there are some people who think some of the ethics gets easier if we take that approach. And I think you've made a good case that neither the statistics nor the ethics necessarily changes. And I think, Rob, you would agree there's some real community issues around understanding acceptability of an approach like that you'd have to sort of wage war with in order to move into that kind of realm. I see a question here from Robert Reinhardt asking, durability, is that a durability of protection at these efficacy scenarios? Any thoughts about that? How does that weigh into the discussions we're having?
Durability is an important factor, and some of these vaccine strategies may need additional boosts year on year. And so the first step of finding an efficacious vaccine is to find out how durable it is, and then what we have to do to keep it durable. And I guess that's maybe Hanneke can talk about how they envisage, should Imbricota be efficacious, but we see waning durability like we saw with RB144. How do we quickly intervene with boosters?
Yeah, that is of course part of the plan, and then of course it's very important to understand at what time you need to give your boosters, right, to not get sort of below protective levels. So that there is indeed some additional work to be done. The good news is, and it's not what this panel is about, is that we see that our antibody titers are pretty stable. But of course, you when you don't know what your protective level is, you need to do a lot of additional research. I do think that compliance to an annual booster, if it turns out to be like that, of course you have the same issue with the long-acting injectables, right? People need to show up for their shot to stay protected. So that's also how do you call it, a more behavioral thing to deal with on how to get full compliance of your customers.
Interesting question from Nina, and I think one that really warrants. Oh, sorry, Carol. Yes, please.
I just want to make a quick follow-up comment. So, in terms of the questions about durability and the relative efficacy, those are all things that really should be discussed, at least at the U.S. FDA, need to be discussed and set up ahead of time before the efficacy trial. So there's sort of probably an advisory committee and a big discussion about what the public health merit would be of a particular efficacy point with the lower bound of confidence interval defined. We've done that previously with other preventive HIV vaccine trials. So they wouldn't necessarily have to be 90%, even though we're now all excited about the efficacy of these other vaccines. But the durability would be decided up front. What is your primary endpoint and secondary endpoint? And that could be expanded over time, in terms of regulatory approval. But those are things that should be defined ahead of time and agreed upon by the agency so that you know what is licensable. Now that's different from public uptake or insurance paying for it, or whether it has a market value. We don't have to make those decisions at the FDA. It's purely public health. So that part is easier. Thank you.
Great. Thanks, Carol. Jeremy, there's an interesting, and it sort of gets to the prep back approach to a certain extent. I'll read it to you. What about a run-in period that provides access to any or all PrEP options and maintains an observational cohort for those who want to stay on PrEP and allows for those who aren't interested in maintaining PrEP to then enroll in the vaccine part of the study?
That's great. It's a version of a design that Ken Mayer, Deborah Donnell, and Connie Kellerman and I discussed in a Lancet HIV paper about a year ago, where the idea is, can we get a population for other means of prevention, whether it's a vaccine study or an alternative means of prevention, of people who don't want to access, at the time it was oral PrEP and will soon be probably injectable PrEP as well. So there are known effective means of prevention, but they're not for everybody. And aside from the great questions, Linda Gale, you raised about bioresistance of the antibiotic over time, there are people who just don't want to take an antiretroviral, whether it's by mouth or whether it's by an injection. And so, can we find those people for whom existing means of known effective prevention are unacceptable, right? And that's unacceptability. And so, what we thought through in that paper is that the idea of saying, oh, do you want to take PrEP? What is that? That there needs to be some mechanism put into place to make sure that that's an authentic choice. So the fact that people actually get to see what PrEP is about, we don't have to force them to take it, but that we actually muster, all the resources we have to say, here's what PrEP actually is. Do you want to take it? Here are the advantages, and actually consider a run-in period where people actually try it so they know whether it's safe or effective. Now that raises different questions. We didn't anticipate the questions about long-acting and a tail that would have to be covered due to resistance, but that idea is one that's really important. And if we could put that together with the idea of people who continue as a cohort so that they'd have some kind of control condition. I'd love to hear what the statisticians say about how we can do that, whether that's an adequate control condition or whether, to be able to make reasonable assessments of safety and efficacy or not. But if not, it still would be a good run-in approach. And that question of authenticity is really there. We just want to make sure people aren't just buying the story that's told by sort of a really mesmerizing research assistant who says, you don't really want that oral prep or that injectable. We want you in this tasty vaccine study, right? We want to make sure that it's an authentic choice.
So Jeremy, I'm going to change that a little bit, and this is coming from Margaret, and to a certain extent, Holly's alluded to this. What if that 3 or 6 monthly injection is not antiretroviral based, but actually is a passive, a monoclonal antibody? How does that change what you've just described? Does it become possible? Do we think, and maybe I'll throw that open, Rob, others, what do you think if indeed the alternative is not an antiretroviral, but passive immunity?
Yeah, I think it's really dependent on whether it's effective, right? So safe and effective means of prevention, whatever that is, we'd want to, and it's, again, the same criteria. Can you actually access it? I mean, what we say the AMP studies come out and we know that, every couple of months, you got to come in for a big infusion. Is that scalable realistically? Are people going to be able to do it over time? There may be barriers to getting an infusion every other month or an injectable. So again, it's all the characteristics of whatever that safe and effective means are, but they get on the table for consideration. And unlike the older WHO guidance that everything that's shown to be ethically or safe and effective needs to be provided to participants, it's impossible. You get injections, oral prep, injectable prep, condoms. By the time you're done, you're hermetically sealed. You'll never get HIV. Nor have a life. But I think what we need to do is consider each of these on their merit.
I mean, I think access to care is a big piece of it, right? Everybody can't go every other month or every 3 months to get an update on their injection or infusion. And I also think that when you're talking about prevention, right, vaccines and prevention, you're talking about otherwise healthy people that may or may not even be involved in healthcare at all up till that point. Their sexual health is the only thing they're going to worry about. And so vaccine is the best case scenario because you can, set it and forget it for however long it lasts. It's going to be longer than long-acting injectables.
So Margaret and Peggy are asking the question around if there was a long-acting antiretroviral, if the monoclonal antibodies are found to work, would there be value for an HIV vaccine research? And I think there would be, because we still need an immune-based durability. So the issue around long-acting ARVs and monoclonal antibodies, they're as long-lived as their half-life in you, and you have to keep on getting top-ups. And so it would be great if there was an immune-based manner of inducing long-term protection that didn't need 6 monthly or 3 monthly top-ups. And so I don't think it takes away the need or the desire to do HIV vaccines, because we do want active vaccination to work, and we do want a vaccine that induces neutralizing antibodies that will be durable, and you won't have to keep on getting your infusion or your injection.
Yeah, that's a wonderful note to end on. Thank you, Glenda. Is there anybody on the panel? We're running out of time. Is there anybody who has a burning other thing they need to say? Please just indicate, and we'd love to hear it. Carol.
So this is a really important panel because I, for one, believe that we won't end the epidemic without a vaccine. So the question is really how to do these complex trials in the face of all these new interventions. And my only message from a regulatory point of view is that there can be a need for these complex trials, and we are open to those discussions. And the important thing is to have a lot of early communication with the agency in the planning. There is an innovative pilot program to have enhanced meetings with the right subject matter experts to work out these models and the assumptions and all that. So it's just going to take a lot of communication with the agency, but we're certainly open to that. So thank you.
Great. Thanks, Carol. That's encouraging and important to hear. Anyone else? Georgia, as a champion for this. What is your last word on us finding a vaccine?
Great, thanks. I just want to extend what Glenda said and say the importance of this question about will a vaccine be necessary, especially if we can bridge it to the pediatric population and implement it as a regular series of our vaccine schedule, such that it's given to a large population before it's needed. That will have a huge impact as well.
And I see lots of nodding heads around there. One last chance to anybody else wants to add a last word. Otherwise, I think we're good. I think you've heard it. We are determined to press on. We're heading for the success we heard this week for COVID. We believe that an HIV vaccine is required to ultimately control this HIV epidemic. And thank you to all of you. You've all been absolute stalwarts and champions of the cause. And we look forward to you keeping it going and others who've joined us this evening. I'll hand over to you now, Roger, to end us off.
Thank you very much, Linda Gale, and I would like to thank you all who participated today in this discussion. I would also like to thank my colleague and their organization who contributed to the organization of these webinars. Deborah Donnell, Holly James, Monica Miller, Stephen Wallace, Linda Gale Baker, Holly Pruden, and Holly Pruden. I would like to draw your attention to similar and complementary work done by our colleague at the Forum for Collaborative Research, as well as EVAC. We're all trying to work towards building research literacy. The next panel discussion is currently scheduled on the 2nd of December and will be on trial of monoclonal antibody. We will confirm these dates as it depends on the public release of the AMP studies. All presentation and panel discussion are available online on the Enterprise website at www.vaccineenterprise.org. We will send you a survey soon to see what you thought about all this series. Goodbye.